All right, this slideshow is on genetics and inheritance, and it covers the readings in human biology for chapter 19. This was one of the fundamental problems in biology, not just human biology, but in all biology, is how are traits passed on to offspring? And why do offspring sometimes resemble the parents and sometimes more resemble a grandparent or a sister or a brother or a distant cousin? This was a source of incredible confusion, and it took a long time to figure out. We'll talk a lot about a person called Mendel, who uh, described basically how inheritance worked by proposing inheritable factors he called alleles, which we can now call genes. And we'll talk about some simple genetics problems and talk about solving them with what are called Punnett squares and pedigrees. And we'll talk about the patterns of inheritance. This was a great, great problem that held people back for a long time when they were trying to understand inheritance. It seemed like it was just too complex, that whether it was a person or a domesticated animal or even a plant, it seemed like it was that traits could appear and disappear kind of randomly, and people were unsure of how it worked, and there were lots of kind of silly ideas that we now know are wrong. And this too also depended upon kind of further advances in biology so that we could understand how all this information that was needed to make a single organism could be carried in a single cell, um, like a fertilized egg. And so, how could you carry all that information to make an animal, a plant, or a human being? They knew from experience that when men and women had sex, that there was a transfer of fluids from a man to a woman, and lots of the ideas around inheritance revolved around this fluid transfer. And they thought it was a blending of male and female fluids. And that really didn't make much sense. And it really wasn't until the 17 and 1800s that we made any progress with this. And there's two guys there. The guy on the left there is Thomas Koch. The guy on the right is Robert Bakewell. They were two Englishmen who were worried about farm production, and they started selective breeding of animals. And this led to an idea that, well, you could control the traits of an animal, even if the traits sometimes seem to disappear, by making sure you bred them for a specific thing. You selected them, so you would always choose the largest cow to mate with the largest bull, and that way the offspring would be very large. Or in the case of Bakewell, there he was particularly involved in sheep, and so he wanted his sheep to have very nice wool, and he wanted them to also be large and be meaty, and so he would select which sheep would reproduce, and he would keep their offspring. And this led them to believe that, oh yes, there is an understandable mechanism here, and they didn't know what it was, but they were practicing selection and controlling inheritance. This was further picked up by a guy named Gregor Mendel, and there he is down there in the lower right. He was an Austrian monk, but really he grew up on a farm, and his great advance was really kind of following up on the work of these other agriculturalists, in that he decided to follow traits of one type of plant, and he would follow very, very specific traits like flower color, plant height, and he picked traits that were very discrete, that it was easy to follow. And then he kept a very good track of the offspring. He controlled the mating of the plants by artificially fertilizing them, so he could choose which parents bred with which other parents, and then he could count the offspring by raising the plants from the seed pods. And from this, he developed two great laws of inheritance: particulate inheritance and segregation and independent assortment. And let me explain what he did. And you can kind of see it there in the lower right. He took plants that were true breeding for specific traits and he bred them. Okay, he called this a test cross. And so he would take these known traits and he would follow them for several generations so that these parents would always have, the offspring would always resemble the parents. So he called them true breeding. And then he would cross um, two different true breeding plants and see what happened to the offspring. So, for example, um, he would take true breeding white flowered plants and cross them with true breeding purple flowered plants. Okay, and so he knew that these organisms carried only the factors for this one type of inheritance, either purple or white flowers, and he would cross them. And what he found was that all the offspring from that cross resembled one parent and not the other. And so for these purple flowered plants crossed with the white flowered plants, all the offspring were purple. Somehow, the inherited factor for the purple flower was hiding any inherited factor for the white flower. Then he would allow these purple flowered offspring to self fertilize. And what he found is then he got a ratio of 3 purple flowered offspring, but then the white flowered offspring reappeared, or the white offspring or the white inherited factor reappeared in 1/4 of the offspring of the purple flowered cross. He then also allowed these to self fertilize, and he found some of the purple flowered were true breeding, that all their offspring were purple. Others had, again, the 3-to-1 ratio of purple to white offspring. And then the white flowered offspring were always produced white flowered plants themselves when they self fertilized. So he did this with many plants, and he found these ratios held up very well. So he used these mathematical approach of counting the offspring, and using peas, he was able to use this approach for I think 7 different traits shown here: flower color, seed coat, whether it's wrinkled or smooth, pod color, um, pod shape, flower position, and the plant height. And so, and he found that these all behaved in the same way, that there was one inherited factor that was able to hide the other, but then the other would show up in this ratio. And so from this, Mendel came up with his conclusions. And one, he said that the inherited factors were discrete. There was no blending of fluids. We now call these inherited factors genes. He called them alleles. And secondly, individuals receive one copy of an allele from each parent. For every trait, they get one inherited factor from each parent. And not all these copies of these alleles or genes are identical. They can be alternative forms. And so individuals can have 2 copies of the same, or they can have 2 different copies of alleles. And so in the case of the purple and white flowers, they could have 2 genes for purple flowers, they could have 2 genes for white flowers, or they could have 1 gene for each. That's heterozygous. Okay. So there was no blending. These alleles would remain separate, or genes would remain separate. And even though an allele might be present, that doesn't mean you'd see it. Dominant alleles or dominant genes were always showed up. They were expressed as physical traits, but other alleles were referred to as recessive. They could be hidden. And so an individual's genotype was the total set of alleles that they contained, but the phenotype was the physical appearance, and that could be different. And so dominant alleles could hide recessive alleles, and recessive alleles or recessive genes would only show up when there were 2 copies present. This made it possible to understand some of the most mysterious parts of inherited factors. That you could have parents that had different traits, and when they would breed, one trait would appear to hide the other, or the one trait would just disappear. So these are Punnett squares here, and they show one parent on the top and one parent on the side. In the case of homozygous parents, they have two copies of an allele. So the purple plant has two copies of the purple allele, which are here shown as uppercase P, and the white parent has two copies of the white allele, which here is shown as a lowercase p, which indicates that it's recessive. It can be hidden. When these two plants breed, you just kind of run the alleles across. They can only transfer one allele at a time. And in the case of the white parent and the purple parent, that's true breeding, all the offspring will be heterozygous. They will have one dominant gene and one recessive gene. If these individuals are allowed to breed, okay, they will each have a 50% chance of passing on a purple gene or a purple allele, and a 50% chance of passing on the white allele, which is the lowercase p. And there you can see the Punnett square below shows their results of their cross. And here, their offspring, one will have two copies of the purple allele. Two of them will have a copy of the purple allele and the white allele. And one of them will have two copies of the white allele. And so that gives us the 3-to-1 ratio. But actually, not all the purple offspring have the same genotype, even though they look the same. Okay, so this allowed us to explain how hidden traits could pop up in unusual times. Okay, and this explained a lot of human disease. One example of this that your book covers is cystic fibrosis. In cystic fibrosis, there's a normal allele C, and a recessive allele for the cystic fibrosis gene is a lowercase c. And individuals who are homozygous, having two of the uppercase C's, will appear normal and be only able to pass on the normal gene. Okay, but if you have two copies of the recessive allele, you'll have cystic fibrosis. And this involves a gene that's involved in normally transporting chloride ions. It's a transport channel, and it fouls up mucus transport, and so these people have lots of mucus in their airways and their digestive system, and so they have a problem breathing, they have a problem digesting. The children are born alive but grow slowly and are quite sickly and prone to infection. And the surprising thing for the inheritance of this was that the heterozygous individuals that had one copy of the normal allele and one copy of the recessive CF allele are healthy. Because they have the normal allele, which is dominant, they appear perfectly healthy. And so healthy parents, normal healthy parents, if they are both carriers, heterozygous, 1/4 of their children can have cystic fibrosis. Okay, and those children need extensive medical care. And prior to just recently, they were all going to die before adulthood. But with modern treatments now, they can reach adulthood. Turns out lots of conditions, genetic conditions, are transferred in this pattern. Another one you might have heard of is sickle cell anemia. Here, the normal gene is for normal hemoglobin, and sickle cell hemoglobin is associated with sickle cell disease. If you're homozygous for this, you will have normal red blood cells, but if you're homozygous for the sickle cell gene, you'll have abnormally shaped sickle-celled red blood cells, and that will cause an anemia. The interesting thing about this is that in cases where malaria is very common, the sickle cell anemia allele, when it's in the heterozygous, confers resistance to malaria. And people that have the normal gene for hemoglobin are very susceptible to malaria. And so in this case, there's an advantage to being a heterozygote as long as malaria is present. You can resist this. And so individuals that have one copy of each allele are favored. And so this is very common in places where malaria is common. However, if people that are move from an area that has malaria, like the Middle East, around the Mediterranean area, in Africa and Asia, then if they move to the United States where sickle cell is not common, the normal hemoglobin allele has no disadvantage because we don't have malaria. And so it becomes more and more common. And then the sickle cell allele has no advantage because malaria is not present. Okay, and so this is an example of an allele that's at least partially displayed when malaria is present. Another type of inheritance that took a little longer to figure out was some genes are sex-linked. That means these genes are carried on an X chromosome, and so they appear to occur more often with males than with females because males have only one sex chromosome. So in this case, two types of inheritance are linked together, the inheritance of sex and the inheritance of genes on these chromosomes. And so the most famous example of this is hemophilia. This is a gene that's related to the ability to clot blood. The normal blood clotting genes are located on the X chromosome. Okay, and so if there is a defective gene for blood clotting linked on the X chromosome, it's more likely to show up in males because males have only one X chromosome. And a normal gene for blood clotting can't hide an abnormal gene in males. So heterozygote females can carry the defective gene for blood clotting and still appear normal, but males can't because they only have one X chromosome. That's the picture there is the royal family, the Romanovs of the Russian Empire in around 1917. Okay, and the empress or Tsarina of Russia carried the defective gene on her X chromosome. Her daughters may or may not have carried it, but her son received that defective gene and he had hemophilia. And so this turned out to be a very big problem because he was the heir to the throne, and so they spent all their effort trying to keep him alive. Okay, and the inheritance of this actually came from the British royal family. Queen Victoria's daughters carried the defective gene, and one of her sons actually died of hemophilia, but not before marrying and passing on that hemophilia. So a very, very serious type of inheritance, and different from the normal inheritance. And so this explains another pattern that people weren't used to seeing. Okay, then even more involved is sometimes there can be more than two alleles for a trait. There might be even three possible alleles. That doesn't mean that a person carries three, but that in a population of people there may be more than three alleles. And this is probably best explained like in the human blood groups. Okay, there are three alleles for proteins or glycoproteins on the surface of red blood cells. There is an A allele for having the A molecule on the surface. There's a B allele for having the B molecule on the surface of red blood cells. And then there's an O allele for having neither of these molecules. And so individuals can inherit two alleles for this trait, and they can inherit two A alleles, two B alleles, two of the O alleles, or they can inherit any combination of I with the A, I with the B, or the O. And so this leads to four possible different blood types because these alleles are partially expressed. Okay, and so these show the blood types there, and these blood types have even more possible genotypes. Okay, and so this is an even more complex system, and it had to wait until we got a little farther along before we could understand these types of alleles. Okay. In addition, although some human traits are controlled by a single allele and we have dominant and recessive inheritance, in some cases, we can track these pedigrees and we can see these things happening in families. And so, in the case, we can see affected individuals and non-affected individuals, and we can track them through what's called a pedigree analysis, and then we can try and reconstruct the genotypes of the individuals. In cases where a trait is caused by a dominant gene, you almost always see that gene in each generation, and if it does disappear, it doesn't reappear. In the case of recessive genes, these pedigrees appear quite different. The gene shows up suddenly, and it can appear to skip a generation. So in some cases, two people that where neither one appears to have the trait, okay, they have children or grandchildren where the trait shows up. Um, and so the carriers here are shown, known carriers here are shown as half one color, half the other color. Individuals lacking the trait completely are unaffected, and sometimes we know a person is may be a carrier, and so we can then notify this. And so the pattern of individuals here is much different, where genes or traits appear to skip generations. Okay, so we have lots of genes where we know it follows this recessive dominant pattern of inheritance. This includes albinism, al- the production of extra fingers, things for color blindness, cystic fibrosis, muscular dystrophy, and on and on. But often, many of the most important genes and most important traits are inherited by interactions from several genes. Okay, and in this case, multiple genes affect the phenotype, and so you see a wide variety of phenotypes. This is like for height or size. These traits often have continuous variation. Intelligence would be another example here, is that there's many genes that can affect this. Okay, and so these were first kind of talked about and understood in the context of agriculture. Mendel helped us see the beginning of the pattern, and then as things have gone on, we've understand how this pattern works.